Hello, my name is Lexis Caputo. Today's date is November 5th, 2018, and today I will be discussing aseptic technique in culturing for culturing microbes for Bio 225. So both lab experiments this week require TSA agar. So in order to do that, I'm first going to prep for both assignments, in which I need a pot. The water has to be approximately 1 milliliter over each tube, and then I already gently loosened each top so that heat can escape, and this will be about an hour process for the water to boil and melt the agar evenly. So the agar has melted completely, and they're now in the plates with the lid slightly off to allow heat to escape as they cool and solidified, ready to be used for experiments. So as I am waiting for the yeast to froth, I already began the aseptic technique experiment by placing E. coli culture into a nutrient broth, as well as epidermidis. And how I did that was remove the lids of both tubes. We put the lids through the flame to sterilize it, and then I had to take 0.25 milliliter of the nutrient broths into the two test tube samples, which are now sitting in bleach solution so that they can be thrown away. And I had to shake vigorously the two test tubes so that the tablet dissolved. And then once dissolved, they were transferred back into the two nutrient broths. And now I'm just waiting to repeat the same process for the yeast, waiting it for 10 minutes for it to froth. And then this is actually the solidified agar solution. After incubating for 48 hours, there's slight turbidity amongst all three samples, with the Saccharomyces cerevisiae with the most turbidity amongst it, and the other two are more translucent. And there is actually flocculent growth amongst all three. However, the Staphylococcus epidermidis has the most defined growth accumulated at the bottom. And now all three of these samples will be used for the part 2 of this experiment, isolating the individual colonies. This is the end result of the 3 samples as they were incubating for 48 hours. It seems as though the Staphylococcus epidermidis was the most successful in colonizing, as it had some pinpoint with very undulate and rhizoid shaped growth. The Escherichia coli had a little pinpoint, and the Saccharomyces cerevisiae had a mix of both, with some punctiform and undulate, and also filamentous shape. It was very hard to actually see where the inoculation loops were actually on the agar from the samples and then back when spreading. I think this was actually the last one that I did, so it might have been the most successful practice of the 3. And maybe if there was more allotted time, these would grow more. So we'll see if this changes for future experiments.